أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. وربنا أتمم لنا نورنا واغفر لنا إنك على كل شيء قدير. ربنا اصرف عنا عذاب جهنم إن عذابها كان غراما إنها ساءت مستقرا ومقاما. رب اجعلني من الصلاة ومن ذريتي ربنا تقبل الدعاء. ربنا كن عنا. السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. إن شاء الله تعالى بإذن الله تعالى we'll be studying week 8 of BIOC17, and inshallah ta'ala we'll be looking at environmental factors shortly. You with me, inshallah? Barakallahu feekum. Assalamu alaikum. Okay, keep talking.
Can I buy something? Yeah, I'll provide it. Now we're back.
Okay. Can I just confirm if Zihan is here? Zihan's not here, is she? Okay. On the way? Okay. So for those that have completed the generation time assignment and they're ready to submit it, you can actually place it right behind my benchtop here, like at the podium. As well, if you haven't picked up your unknown assignment sheets for your flowchart and data sheet, they're actually down at that end with the slides. Because of timing for today, I just want to focus and concentrate on this, the slides and your incoming presentations. As you could see from the PowerPoint, it says present as a group, presentation requirements formal lab report. I'll talk about this briefly, and I don't want there to be any form of pressure on you guys. I want you guys to, feel confident with the results that you've had thus far. And hopefully you've seen my inbox message last week, because I'll give you a hint as to what you can kind of expect for your results. Anyone missing, by the way, in terms of updates from the past, any outstanding questions from the past or even now? Everyone, if actually I forgot to mention, anyone wants to essentially, obviously before the end of today's lab, I would need the, what do you call them, your generation time sheets here. But if there's any final calculations that you need to do, just let me know. In the meantime, actually, once again, any questions? Any questions? Orient here. Everything okay? You guys are okay? Yeah, okay. So congrats, by the way. You guys are almost 80%, right? A-level grade, hopefully, into the practical. So I'm just going to briefly go through some things, the slides for about 10 minutes, and then we're going to try to go through the material in due time. As you're aware, we're dealing with environmental temperatures, and this is actually one of, like, I think many of you hopefully will like this practical. In terms of the exercises for today, we'll be looking at temperature, salinity, and pH. And many of the tubes that you have are actually in the middle bench, and hopefully, inshallah, we'll look at them, see and observe, and be able to accordingly take as different groups today. I want to also mention that we may be splitting up, so let me just see some things. Okay. I'm just keeping in mind a few things for later on today. So for essentially analyzing and based on what we've seen from the lab manual, there are different categories of and classifications of bacterium. By the way, Cedric, can you see the back end? Yeah, you can see the slide. Okay, everyone can see the slides, hopefully. In terms of the bacterium, we have different types, different classifications. We know thermophiles, mesophiles, psychrophiles, etc. And that goes from, living in high temperatures to ambient temperatures to cold temperatures, indeed. And essentially, the goal is that you're going to take a loop full. Let me just show you, orient you guys through the tools that you're using today. And actually, if possible, I recommend recording this so that way you're not lost. Because we're separating like for each exercise or each set of exercises, it can be different groups and different responsibilities. So just bear that in mind. Mainly for exercises 1 to 3, you'll be in your regular groups of 4, 3 to 4. But for exercise 4, you'll be dealing with groups of 6. So we'll likely be splitting into quarters for today. But inshallah, briefly, I'll highlight these things. You have nutrient broth here. Each group of 6, so remember, specify 1 temperature. Each group of 6, each group will be taking 10 tubes, right? And that will reflect the 4 different they were labeled. In both you have them in your manual as well. But you'll have essentially 10 tubes each group with 4 different types of bacteria and a rep of your control. Control, you don't add anything essentially, so you'll leave it as is, serving as a negative control without any bacteria. And then you'll do that because that's technically 4 bacteria plus a control. You'll do that twice. So you have 2 reps. Now I'll give you 10 tubes. Across the temperatures that we'll be dealing with today, I'll talk about that in due time because I want to orient you guys clearly. But we hopefully will be able to look at, for each of the groups, a specific temperature. We have, I believe, 5. 5 different temperatures. Okay. So we have 6. And just for context, a lot of this is a bit static, so don't get confused that some things change up essentially for the manual. But nonetheless, you guys will be separating. Now I'll provide you details as to what you be doing in your respective location. pH is here as well, and you have 8 tubes. 8 tubes for pHs. pH 2, 4, 6, 8, and 10. And in that regard, we'll essentially have 5 groups. We're all going to you guys into what you'll be doing. That essentially you have the bacterium and you will follow the same reps without the controls. Because technically for control you have to have pH of 7, and we don't have a pH of 7 here, or alternative is to not have bacterium in those respective tubes. Another thing is that salinity, is essentially going to be done in groups of 4. The same regular groups that you have, and you'll be taking 1 of each salinity. So I'll talk about that briefly, but you have the bacteria, you have the label, and if you need help support in knowing which bacteria am I using, then I'll help navigate you guys. But you have the inoculating loops, the sterile ones, so bear that in mind. You want to take the larger end, which is at 10 microliters, when you're doing your inoculation. So recall a septic technique. You're going to be flaming your tubes, right? And bear in mind, the ones that are liquid, right, are the ones that we're using for the first set of exercises. But remember, you have a pellet. So each person in your group should be very mindful and particular that you resuspend. And you should, by the way, just close fully before you do so. Otherwise, you don't want it to spill out and get back to your lungs. So you can resuspend in this regard. Do it for all your foods in advance, and then accordingly, remember a septic technique and potentially use sterile food, sterile inoculating loops. Don't need to flame them. You don't want them to cook. But essentially, you'll take 1 loop full, place it inside, gently resuspend, and then we'll accordingly return them back here for the respective tubes for salinity and pH. Before you move further, I just want to really highlight something very important, and that is to please label your tubes, because we're not only doing this for specific groups, specific exercises of bacteria and concentrations of pH, temperature, salinity, etc. But it's important to bear in mind that we may also have other groups, other practicals in similar locations. So please make sure. I'm actually going to write this down so you can just remember this. And in thermal death time or thermal the decimal reduction value exercise form materials here. But I'm just going to highlight this so we're all on the same page. You can do this on the tube itself. Our practical, right? Let's see that. And then the exercise. Actually, instead of exercise, put the well, you could say both. If let's say it was exercise 1 and you're at 30 degrees, then you'd specify that, specify the bacterium, this E. coli, and specify the rep, because you're doing 2 reps, usually for across the different conditions. And then you could place the condition or the rep that you have, and then follow it with the date. The date is so 318, you can go 25 or 318. And that should be it. But it's very similar, replicable to the other exercises. And with that, any questions? Can everyone essentially sterilize their benches? Has everyone done so or not as of yet? If you haven't done so, you can begin this now. And I'm just going to briefly mention, oh, perfect. So the bacterium are highlighted here. So you guys should hopefully, no problem, no problem. Do you need any help?
No.
Okay, no problem. So with this in mind, start off with exercise 1. My advice, I want to see if we can make this efficient. If you can, this is my recommendation. And I think I shared this. If I haven't, I'll share it with you guys now. Because there's so many exercises with similar steps in mind, try to actually separate in pairs. So, for example, a group will, so within your group, groups of like 3 to 4, have someone focus on pH, or a pair of you focus on pH, a pair of you focus on osmotic pressures. And then for exercise 1, try to do them in sets accordingly. You can also separate, like you could do exercise 1 as a pair, exercise 2 as another pair, and whoever finishes first can do exercise 3. But the aim is that you will use the bacterium as you could see here, and we'll follow through each exercise in due time. My goal, objective is that we should try to finish all 3 exercises in an hour, because it's not too difficult. You're dealing with very similar bacterium as you probably have seen before, and many of which that you've viewed through the manual as well. Yes. So that's another thing. Are you talking about the temperatures? No, so as I see now, there's only one group that would make a group of 5 essentially, because we have a few members that are not present today. Let me see if I can make something better. Okay. This is what we can do. See, the thing is, technically, I have 6 columns for 6 groups, which is why technically I have it as such. But at the same time, if we look at exercise 1, there's only 5 temperatures, right? So I don't really. I would recommend doing a 6th temperature as well. So let me just highlight for you guys, and maybe make it a bit simpler. If we go it down. Okay, so 10. So this will also help orienting you guys. You can also use Okay, never mind actually. So orienting you guys for the temperatures at least. We're gonna be at 10 degrees in the fridge. So in our location, top left side for the fridge. And just remember your locations as a group so that way we can orient them correctly. But we'll place at 10 degrees there, 20 degrees at room temperature, which is gonna be behind where we used to have the endospore stain test down there. So in the fume hood, groups. Yeah, feel like last time we had 6 groups, that's why. So, okay, nonetheless, 30 degrees will be at room temperature, actually technically in the bottom incubator. And then it will be on the top left side. 37 degrees slash 39 will be on the top left of the first top incubator. And then 60 degrees. When you're here, folks, just let me know whichever group you separate to. It'll be at the very top. And someone will get to get hurt return to this one. So we'll just clear that. Yeah, any questions, any follow-up questions? Yes.
It's this process too that, yeah, I told you that we're gonna be assigned to pH. It's like, keep like that.
pHs, I mean, yeah, we can assign them. Yes. So let me now. Okay, so for temperatures, we'll keep it as is as before, group of 6. And I'll follow, so okay, group 1 will follow with the fridge. And fridge is right there. Right? Group 2, you'll be doing room temperature inside. Group 3, you'll be doing incubator, on incubator top left side. And then group 4, you'll be doing the top incubator, just where we normally place the plates. Group 5, you'll be doing with. We once upon a time had another incubator. I want to do. I want us to put out. We'll put it at 50 and group 6 again. So that way there's more diversity in that regard. So don't worry about significant changes. And then when it comes to pHs, I wanted to start with this because last week we did 2 sets of plates for control and parameters. So you'll be putting pH 2, pH 4, pH 6, 8, and 10. So just bear that in mind. And then, so, this is what we're gonna do for temperature, thermal depth time. You should have. Okay, does that make sense for exercises 1 and 2 or no? And then we review the results, hopefully next week, inshallah, about the turbidity and quantitative measurements from the Spec 20. That's for exercises 1, 2, and 3. Now, so for salinity, I'm gonna separate you guys into those things or those 4 groups. So essentially, split yourselves into halves. Or like when you're at that step, let me know because I could separate you more accurately in case someone else can complete it. Then you could essentially put so we'll have 5 here and 5 on this side. And I'll talk about that in due time. I just want you guys to focus maybe for now, exercises 1 and 2. Recall you're just doing it in your groups. And then when your group is ready, remember before you begin aseptic technique, label your tubes as I've shown before. Make sure as well that you know the bacterium that you're using and then you're following aseptic technique with the tubes as well. When you're done, essentially I'll try to put racks into different locations so you can place your tubes for the temperature in the right location. And then the other two exercises for pH and salinity can be returned if they're labeled. Remember, you need to label them. Very, very important for your group, the exercise, the bacterium, etc. And it's very similar for osmolarity in that though you have osmotolerant tolerant and non-osmotolerant tolerant bacterium, i.e. those that are able to survive at higher or lower, like essentially changes with salinity, and then non-osmotolerant are those that are generally more sensitive to changes in salinity, like sodium chloride concentrations. Um, and then pH, acidophiles, more on the lower pH end. They have alkalophiles or basophiles, which are on the more basic end. And then you have neutrophils that are more in between. There are other categorizations with some similarities, but the point is, I want you guys to be able to do this with success. I'll put a timer for 10:40 to be done all three exercises. Let me know though if you need help. If you have questions, more than happy to help in due time. But you can get ready. Remember, benches need to be clean. And when you have your tubes, make sure that they're labeled. You can also grab the inoculating loops. So have two members of your group here. And in due time, just remember to have everything labeled. That's okay. That's okay. No problem. Really appreciate it. Uh, you're gonna use the larger circle. Oh, no. Grab the one with six. It's okay. Oh, you have your name though, right? Can you just put your practical number just as well? That would be helpful. I'll give you a pen. Pen is right here. I really appreciate it. So folks, I just put the timer. And if you need a bit more time, let me know. Some are spilling. Make sure that your tubes are closed properly. As you use them and before you use them. Actually, folks, regarding the unknown assignment, leave that aside for now because I want you to be able to, at the very end of today's practical, at least give me your generation time assignment and your unknown. So, everything okay? What's happening? Folks, make sure you place your tubes on the trays. They are not solid agar, so they can't spill. So,
Okay. So because we have a limited supply, I'm gonna ask the lab tech if we could get more. If we can't get more tubes, we're actually gonna have to be supply. Like how can we buy food today? So just continue as you are. For the groups that have actually the drop,
We're gonna use page 4. We need a page 2 of the page 4. That's it, you have page 4, yeah. No. Well, I don't know what. Yeah.
So folks, remember to label your tubes. Make sure that once again you don't have them on the slide. Have them in your racks accordingly, and inshallah they will school. I do want to highlight as I get extra straight.
Container to put them in and put them like in their proper place? Is it just the tray? Like, do we keep them on the tray for like at the end? Once it's all like the pH where we inoculated our bacteria, and then it says place them in a container and then incubate them? Is the container just the tray?
Yes.
Can we just use the same one pH, like the same labeled? When or should we grab a new?
There's a new one of the we have extra trees on there.
We'll leave that.
someone open that? Was it closed? So that's what normally happens when you have a private patient panel. Folks, I just want to take this opportunity to say something. Please do not think of it the wrong way that I'm like not fully involved with you at any times. I say that because for one, things change quite dynamically in the lab. See, we're now likely being 5 groups, right, instead of 6. The other thing is that I'm not, to be very honest with you, I appreciate it, but I'm not getting enough resources. So I just want to like let you know I'm not in any way, shape, or form trying to like jeopardize your lab performance. Do your best and leave the rest with us. That's all I can say. And I hope you guys do your best in this lab. Just ask me if you have questions, if you have problems. And I wanted to see you all some. I'm very so just let me know if you have problems, concerns. If you need me to clarify anything, I want to talk. So, folks, how's it going, by the way? Are you guys doing well? Please check the technique. You guys you need to stand your back here, folks. Before you speak anything.
Is that a pet I do? Yeah, for George, he said it like sensitive that
Maybe Staphylococcus. Yeah. Okay. Okay. Let me actually answer this now. So folks, I'm just gonna clarify things. Just to confirm, group 3, are you guys doing the temperatures or you just skip that?
They're going to.
But you don't have liquid. How many? What happened to the ones that were like? How many? But I had a clear one. So, folks, just to repeat this, clarify this once again, for experiment 1, you'll be having the temperatures that are available here for 10, 20, 30, 39, 50, and 60. And just remember, please label your tubes accordingly. And if you have questions, let me know. The bacterium include for exercise 1, your control, left empty, E. coli, B. sterile thermophilus. And by the way, the suffixes should give you a hint as to what classification they're part of. Micrococcus pyophilus, which also gives you a hint, the phyllis being attractive to cryo cold temperatures. So that should give you a hint as to what type of bacterium it is. And then you have B. subtilis. And each of those bacteria you'll be looking at to be able to observe their turbidity results. Folks, let's just pay attention just a bit. I want to make sure you understand the relationship, because by you being able to analyze the turbidity, you're essentially observing population growth. And so based on the differences in their thermal ranges, you should be able to then deduce, even with the suffixes, hopefully, inshallah, what you expect then would be their maximal or optimal growth regions, and where they would essentially at the right temperature grow best. And that's what you'll see qualitatively next week, inshallah, with the turbidity, changes in cloudiness, and quantitatively with the spec 20 absorbance readings. And then it's very similar to pH. Now that we have only groups of 5, my goal is that you guys stay as I mentioned before. Okay, yes. So pH 2, group 2, pH 4, group 3, and then pH 6, 8, and 10. And then for osmolarity, you guys will do it in your respective groups again. Yes. Don't worry about the controls. Don't. You don't have to. Because technically the controls are literally left for the temperature, right?
Because if there's stuff in the control,
But the media itself is gonna very likely stay as is. So I don't like. I'd rather you have something to compare with, like because the 2 reps. Yes, and we'll see from the other groups as well. Your temperature is 39. Okay, incubator. That's okay. Trust me. Don't worry. For let me just see. Bacteria, right? Can I just see? Okay. Give me one moment.
The disposable ones, they go in the wet bag.
All 4 words. Anyone started exercise 3? Any group started exercise 3? So I'm just gonna make an update to make it effective once again. Exercise 3 and 4, I want you to be in groups of of 6, 5 to 6. So you'll be separated into quarters. You'll be one half, you'll be one half, and essentially there'll be 5 of you on that side and 5 of you on this side. I do want to highlight as well that way you'll be able to go straight ahead from exercise 3 to exercise 4, and you guys could split up accordingly if you want to as well. But in the meantime, focus on exercise 1 and 2. And then when your group is at exercise 3, because you'll be dealing with just one osmolarity, I recommend returning back the same tubes that you had from before to place it where they were initially. So hopefully if you have them already labeled, it's okay with like the concentration of NaCl, just return it back. So that way all quarters can accordingly get the right concentration of NaCl. It will be going up in the way that we've talked about before, 2%, 5%, 8%, and 11%. Temperature, exercise 1. I have a tray already inside. If you're focusing on exercise 1 and 2, just stay focused. Exercise 3, don't start yet. We're gonna essentially shift to those groups of 5 to 6, or into those 4 groups accordingly. Well, please make sure just once again to have your tubes labeled, because other practicals may also have their tubes in a similar tray, and we want to be able to identify them. Use the points shown here, but also from the lab manual to just elucidate that, okay, this is our group, this is the concentration or temperature that I was using. We're done 1 and 2. It's right. 60 degrees. But when you guys are, don't forget to resuspend before taking inoculating loops and after in your final set of tubes. I've already placed some racks in different locations. When your group is ready, take out those tubes or the rack itself, and then you could thereafter place it in. You could put it directly with the trigger. Yes. Is that your trigger? Just to confirm? Like, did you get it? No? That's okay. You can do it. Everything is labeled. The tubes are suspended and everything. It's gonna heat up. It's okay. Because it's also gonna do. Yeah, it's not. Yeah, you can put them in. I would place a blue dot here, so that way you can select. There was a blue dot. You guys can place them. You can switch them there, and then you can still keep that one. Place it in here. Because it will actually like this.
Yeah, so I'm like, now there's a chance that I was, yeah.
Well, whoever is doing exercise 3 is gonna have the 8% for NCL back here, and the same for the remaining 2%, 5%, and 11%. Essentially, we're going to separate into quarters, and I'll give you descriptions when you're ready. But in the meantime, try to be done exercise 1 and 2 in the next 5 to 10 minutes. Yeah.
Where do we put it?
Yeah. I don't want to. Folks, just a reminder, when you're done exercise 2, pHs should actually be brought to the middle bench. So bring back the pH tubes that you're using, and then exercise 3. Inshallah, what I'm going to talk about now is splitting up. And what we're going to do is that have 5 members. By the way, who's closest to you?
Yeah, we'll just take.
Marissa? So you guys can, so if that's okay, when it's due time, you guys can already start actually exercise 3.
Okay.
So you guys, when you're ready, I don't want to inconvenience anyone, but I know we have maybe in the meantime, the next few minutes, I just want you to get prepped to finalize the remaining 2 exercises. Thermal death time we'll talk about afterwards, but I want you to, inshallah, focus, complete. Let me update the timer so you guys could see where you should be done. When it reaches 25, so essentially in 6 minutes, I want you guys to be done. Exercise 1 and 2. If your group is done, remember, place them back into the correct racks, right? There's some of them that are present around for the fume hood, for the fridge, for the 2 incubators, for the 50 and 60 degree water baths. When you're done that, if another set of your group members or your pair is done the pHs, they can return them to the middle bench. And then for the 3rd exercise, you'll be in the groups that are split up. So you guys will split up. Group 5 will primarily be on half on one side, half on another. And then we've already talked about folks for exercise 3 and 4. Just bear in mind, Marissa will be on this side, and the remaining of you will be for the first group. And that'll be key for 2% for group 1, essentially. Group 2 will be at 5%. Group 3 will be at 8%, and group 4 will be at 11%. You will be doing all bacterium, which will also be nice as an assay, so that way you're able to control at the specific salinities that you're working with. But I just want you to bear in mind that you do this resuspending. Be careful and mindful that you have the bacterium and that you labeled the tubes accordingly. When you're done, you can return them back to the middle bench, and I look forward to placing them into the incubators when appropriate. Any final questions for exercises 1 to 3? There is essentially you're doing the same reps. You'll be doing 2. Actually, they're actually here. E. coli, Staphylococcus cerevisiae, and then Staphylococcus epidermidis. So if you see the slide, we'll see them right here. Nope. Did you use them each time, or do you? Okay, let's see. If they're all
You don't do this one. Okay.
You grab the tray for exercise 3 and then they put it back into that. Make sure you Fine with that. I'll answer your question. Um, I believe we had 6 tubes, right? So So what you could do, based on the splitting of the groups as I've talked about, you have for exercise 3, how many, just to confirm, how many tubes are on track? 5? So what you should do is essentially have 3 of the 3 different bacteria. And you could leave one as a control and one as one that you return back at the very end. So leave essentially 2 serving as controls. One will be used for reuse for other practicals as well. But you could return them back. If you made a mistake, right, then let me know, and then that could also be considered. But generally speaking, remember that for exercise 3 and 4, you're splitting up into quarters. So just remind yourself that that's gonna be also impacting where you see your results for next week. Yeah. John, just to also reiterate this, you have about 24 minutes to complete all 3 exercises. I just want to repeat this one more time for exercise 3. You have 2 controls, one that's labeled, one is unlabeled. The reason that you're going to have one unlabeled is so that another group for future practical can use it. If it's already labeled, just cross out that line for one of those tubes. Or you could take out the tape. The other thing is that, remember, exercise 3, 3 different bacteria, 2 controls, one labeled, one unlabeled. Essentially, it's like 3 bacterium, 1 control, and 1 left as blank. So essentially, it could be used for other practicals accordingly. Yes. Any questions? No? Okay. Everything okay? Everything okay? Yeah. Are you guys, someone's working on exercise 3? They are? Okay. That stuff would be better. So I'll show you, give me just a minute. And I just want to make sure I go over other things. Let me see if I can grab it. Okay. You have anything?
We still have this. Should we not? This is a loop. Sorry, I'm sorry.
So we may have extra groups around if you're missing any. Seeing if other groups have. There should be 9 packs. So hopefully you should have enough. Okay, okay. Maybe some other. Don't forget the sterile plastic loops. You don't flame, right? Because you don't want them to also degrade. But now let me, if everyone's okay, we could talk about exercise 4. Everything okay? Yeah. Okay. So let me just show you the screen. So it's actually like a sequel to our viable counts for generation time assignment, right? So like think about you doing what you've done before, but we're going to be starting off with E. coli, which I don't know if I have here. Perfect. So we have 2 blocks. They aren't labeled, so I'll confirm it. The E. coli is here. We have the NA plates. And so briefly, let me just highlight what you would be doing. You have 1 ml of the E. coli, which is in that small tube that I'll resuspend. And then we'll be placing them into either of the sterile nutrient broth flasks, one at 50 and one at 65 degrees. And so I'll essentially be using the 2 benches that were, or the 2 warmers that were used earlier. And remember, so once again, you'll be in into those quarters. You'll be split into quarters. Once again, the same quarters for exercise 3. Folks, let's just remember we're paying attention. If there's spills, let me know, like major spills. But generally speaking, make sure that you're fully cooperative, because exercise 3 could be done by a third of you and exercise 4 could be done by another. So you guys could split up even into halves accordingly. But remember that you have exercise 3 done by 1 quarter, or sorry, you're split already in 2 quarters as before, and then half of you could do exercise 3 and another half could do exercise 4. I've spoken about already exercise 3. you're doing instead all organisms with a control. So just bear that in mind. One that's unlabeled because it could be used for other groups or reused if needed. Exercise 4, though, is a bit more nuanced in that it also gets back into the math. So you just want to bear in mind it's like an inverse generation time graph or curve. And so what you're doing once again is thinking about the plaque assay. In fact, you did serial dilutions, right? And so this is what you're going to be doing again. Remember, you're grabbing in tubes. You likely have already a tray. If you need more trays, let me know. But you need to label those tubes, remember, of serial dilution, so that way you know and you could track correctly the dilution factors of your tubes and of the plates. Your 5, if not 6 plates that you'll be using today. So you could grab 6 plates into those quarters that you are of right now. And remember, following the steps as you see here, you're doing 1 mil essentially transfers or aliquots. I'm just wanting to see if we got there. For exercise 4, you have 2 options. You can go with the glass, the glass pipette chemistry. You show here. Just bear in mind once again, you're using the right one. And this is better. Like, of course, the microbiologists are more accurate, but sometimes it's better to be a bit crude and being able to take measurements with larger instruments. So see if you could use this. They're more useful as well for specific concentration. So let's see. Yes. So you can use the same. If you're doing it correctly, you can use the same canister following each 1 mil dilution. So we have 5 different. So essentially, you have 5 different tubes. Hopefully, so you have 5 different tubes with 5 different dilution factors going 1 mil at a time. It's 10 to the minus 1 relative to your original plot. So hypothetically, if the concentration was 20 micrograms per mil, what would be the concentration of your first dilution? You would know? It was 20 micrograms per mil. It would be 2 micrograms. So it would be 2 micrograms per mil, essentially divided by 10. And then you keep doing that until the end, and you would essentially bear in mind the units that you're using. After that, because it's 1 mil each time, you're thereafter taking 0.1 mils into the place. Meaning essentially you're doing an additional dilution thereafter. So it becomes from 10 to the minus 1 to 10 to the minus 2. 10 to the minus 2 to 10 to the minus 3. 10 to the minus 3, 10 to the minus 4, 10 to the minus 4, 10 to the minus 5, 10 to the minus 6. And so essentially, you do that for your plates. Remember that you're now in different groups, so just bear in mind, if it helps to make it easier, use letters, group A, B, C, and D. I don't mind if you use numbers, but I want you to be able to coordinate this because we've gone with 1, 2, 3, 4, 5, 6 before. Now that you're in quarters, you can split up into letters accordingly. Any questions?
So in the experiment, like in the lab manual, it talked about different time points.
Yes. So let me just see.
I was with the science. We were assigned Celsius and chloride, and we were assigned time 5.
I'm 5. So okay.
There was time 0 and then.
So this is a great question. In the groups that we'll be designating you shortly, and I just want to make sure, are we done exercises 1 and 2, all groups? You done all both? You done both? Group 2, are you done both? Okay, and group 3 as well. Okay, so and that's okay if the group wasn't able to do it for particular reasons, but we're going to now move to exercise, talking about a bit more about exercise 4. Hopefully you guys are almost done exercise 3 as well. Can I have a show of hands of groups that are done exercise 3? Everyone's done? Perfect. So now everyone's ready for exercise 4. This will also make it easier for splitting up. So it makes it a bit tricky, but I'm going to need a separate timer. Okay. So I'm going to need to do things a bit controlled. So I'm going to ask if folks that have questions, if they could save them for afterwards, because this is timed for me as well. And then you guys will also have to bear in mind. So if you look at the slide or on the sheet right now, page 116 shows the separation of our groups into quadrants. And so group 1 would be 50 degrees of E. coli at time 0, meaning essentially, and this is for time 0, and everyone could also have a timer for this side because it'll be time 0.5 at 5 minutes, I think. Yeah. So the idea is that as soon as I add in the E. coli or the particular bacterium into either class, that's as soon as they do that, you're going to be taking out and essentially making this bacterium. I'll actually do it for you guys to make it easier because yeah.
This is very like that you will find the reading of it at time 0, and then you'll like, and then whoever is at time 0 will just take the 1 milliliter and then start using it from there, like serially. So they only need to take from the lab.
The second time, can you clarify because time 0. Oh yes, at T0 and T5. Is that what you're saying? Yeah, yeah. So that's correct, but I'm just trying to see if I can get something done for you guys as well. In that, this technically has to warm up for a bit of time. So let me go over a bit about yeah.
Wait, are we group 2?
You're group 2. Yeah, so you're doing that T0. T0, T0, T5, T5, 50 degrees, 65 degrees. In the meantime, you could get ready your still dilutions, just making sure that you're labeling them. You could take out your 6 plates, labeling them accordingly. I'm just going to highlight that right now and show you the slide just to reorient you guys. We'll have to wait a bit for it to essentially warm up the agar. Actually, everyone knows how to label their plates, right? Exercise, group letter, our practical, the dilution factor, and the time point / condition of your bacterium. So let's just quickly go through. Yes, okay. I don't have yes.
When I see the device on the glass, what time 0 you need to place 0.1 right away for the 10 times 1, and then we also take the 1 milliliter to us
Yes.
That's
That's sound like that's fair. The thing is though, so okay, this will take a bit of working. Reason being is that I'm also going to be dealing with a microplate reader, so I just want to highlight this. For the first 5 minutes, it will be relatively focused on the quadrants or the groups that you're set in. But for the remainder of about 20 minutes time, so likely around 11, I'll have to really be focusing on the microplate. So try not to ask, like, just bear that in mind. I will highlight a few other things in the meantime and take out actually your your plate. So in the meantime, you can get ready for the presentation. I want to highlight this once again. I know you guys are amazing stewards, scholars, microbiologists, and you're interested in understanding the concepts that we talked about. But I want to see if you guys are able to apply the knowledge, perhaps in an applied setting, essentially. And I'll describe what I mean time. I actually wanted it to be open. Okay. To be honest with you, that's the reason why I'm concerned about the UV, and you'll see what I'm talking about. Everyone saw my email last week, by the way, Friday, yeah? So I talked about essentially some of our results not appearing as they should have been. I didn't want to like divulge and I don't want anyone to get upset and stuff because there's technical things to bear in mind. There's safety for all of them. I consider it as a top priority, and I don't want anyone getting in direct contact with bacteria, whether they're stationary or mobile and essentially airline. So now here's the thing. Normally for these experiments, you want to have the lid off, but there is even with the experiments I've done, I've seen essentially poor results. So what I'm thinking is that there's a few things. One is the starting culture. Some of you have asked me about this before because you would have saw your agar, the solid agar with very thin concentrations or thin culture of your bacterium, and that could impact the effect that UV has at the initial time points, via 30 seconds, 33 minutes, all the way to 30 minutes for exercise 1 from last week. So what I mean by this is that you'll probably see your results and you'll be a bit confused because all of them look positive. And you won't really see changes in UV impact. So what I'm going to be doing then is essentially looking at providing you guys for your FLR, and actually for your presentations, I just want you to be able to describe what you see, what you would have expected, and essentially answer questions that you see from the line on you, because there are a few for the last bit of this week's exercise. So let me just see. Any questions, folks, in the meantime? I'll be giving you. So this is Microbiology roseus, by the way, right? So you would have seen a reddish tint in your bacterium. I'll just organize your plates accordingly. In the meantime, you can start getting ready actually for your presentation. Let me just see. Yes, so now this is what I'm gonna do. I'm gonna try to take out your plates. I just want you to start thinking, describing, okay, who's gonna do what? Intro, results, discussion, following what I've talked about in the past, right? So think about UV and being able to describe the refill effect that you could provide. And then contrast that with the repair mechanism, dark and light, and that's reflecting the purpose of today's last. Right? I'm just going to organize your plates and then see if we can cover your result.
Are we doing both exercises?
Both exercises. So I would recommend splitting up. In fact, because you guys are already groups of 4, okay, continue, Ashley. Continue as you are. And I'm just trying to see to make use of our time remaining setup for today.
That's it. That was just for. Mine is probably the one. Yeah, usually it's so two people do one. Do you guys want to do? Do you guys want to? Sorry, the one. One of we'll do one. Okay. Yeah.
Just folks, if you guys want to grab your plates, we stand now. I'll be on, like, I just want to also highlight some of them you may have to double check. I can't fully discern all of them. So if you can, based on your group number, identify also with writing which one's yours, and you should be able to, like, from the 6 or 7 that you have, then you should be able to hopefully identify. I'm going to grab the ones with aluminum foil shortly. And those are 5 from the light RC. For the dark. But actually we have some for the light as well. So, let me just place them, and you guys can. Dark. four that are like, or essentially the pale incubated ones that we planned from before. Yeah. In the meantime, I'm going to be starting exercise 4. So I'm just going to ask if you can have 2 members of your group as the rest of them organize the plates and like confirm the results for observations. 2 members of your group can come to my side, the quarters. Remember, you have group A, B, C, and D. So 2 members of, or even 1 member per group can come by. And inshallah, we'll look at being able to control your results accordingly. I can have food. Oh, there it is.
Sorry.
Do you see anything outside?
No.
No? Is that for the antimicrobial?
Exercise 1 from last week. So what you brought out was just exercise 2. Yeah. It's okay. You do that. I'll get him out.
Are you sure?
Yes.
I really appreciate it.
That's
There's no bacteria in the what is the maybe are they controlled and they're not?
Very much. Think we can make some. We'll have 5 minutes at max to do your presentation, so please bear that in mind. Try to highlight the main results. Very limited discussion for other points. Just mention the intro, results, and discussion accordingly. Saline tubes will be for dilutions.
Yes. Are you going to normally do that or?
That's for the microplate reading. I should. What's your question? For the class, you good? I just need
Yeah, saline makes for the diet.
it should be clear here.
So we
Every- Oh, look, you're fine. Actually, will you just hold them up? Good. I'm gonna ask for the quadrants now. If they could start getting 2 members, folks. 2 members from each group should come by for exercise 4.
I'm sorry, I'm new. Sorry, both the 0.1 and the 1. If I have, with my sitting like this, I need 1 value of it, and then I need 0.1 on it.
Okay. You may use it directly from your plate. Oh, okay, I see. Then I have to coordinate just a bit more.
No, there's 2 times. I got
Folks, can I have a member from group 1, 3, and 4 with their plates and tubes? Yes? Okay, perfect. I need group 3 and 4 as well. Is it?
Yes, of course.
Group 1. That's you? Yeah.
I really
You
appreciate
first.
it. Then I'll use the microplate. And
No, it makes sense. Of course. Do you want the 3 first or the 2?
Plate is available. Does it, just to confirm with you, does it say about placing it from the original flask or from the tube itself at 52?
See, that was I was confused about
believe
that
it's the
because I think it's the flask. I think you inoculate the flasks and then we take from the flasks.
Now, next question is, yeah, okay, so I'm gonna
We
do
only
that.
have 2 flags, right?
That's okay.
Okay.
This is 50, so I'm gonna do this. And it's,
What group are you guys?
this will be okay. This will be okay.
What group are you?
Oh, one he's done.
So he's doing it in order, so he's gonna do group 1 for 50. And then I'm assuming
Put it in.
it might be easier if you do per temperature, right? So 1 and 3.
Groupings for one, to do this a few times. Sorry. 1 and 2 can be. I realize it said it's all 4 groups.
Yeah.
Um,
At the bottom. That's, yeah, number 6.
We can spread that, right?
This is like
Okay. Thank you. Julia.
a bit pressing. Definitely, one of the places we have can you take it all the way from I'm sorry, it's a bit
Labeling, you take your time.
it's just okay. If we take a fine piece of tree,
So the time points for 50 are gonna be different.
yeah. And it's kind of 40 minutes from the stop as well. Let's go on.
I can set a timer once you've done this for 5 minutes and then
Really,
the 1 milliliter or the point.
the one. Because I'm gonna place it into the
Oh, yeah.
sauce.
And then in the You want me to set the timer as soon as you put it in or when you do the microplate? Okay.
3, 2, 1.
Good.
So I'll give it to you. Actually, can I go with the yes, you could go with the if you could go. If you don't mind, can you keep it going? You keep it close by to me.
Yes. Yeah.
Because I'm going to use it for 3 minutes and keep it so for 15 minutes. So I just figured that's it.
Let me just go get it so they can spread it.
That's it. Thank you so much.
Yeah. This is
Can you make sure that you let me know what the work inside writing? The serum. So she's spreading the one right now. This is the serial. Yeah, this is already. It's already. That's already.
Can you please come here? You need to have your plates ready.
Yes.
And the same for group 4 shortly. What's the time like?
1 minute. Yep.
You 4, can you please come? With cube and your plate. Thank you. But it's okay. that should be okay. I'll try to keep track of you. Really appreciate this.
This is the microscope. Yeah, good look. Yeah, I think this is one, right? This 1,000.
Yes, Jim, had a question? Here, was there a question? So this is a bit of a fun fact. As you guys are doing this work, I'm dealing with a microplate reader, and you can actually tell over time that the bacterium are, lysing, and so eventually you're likely to see what's shown there, a decay of bacterium over time, and the turbidity readings thus over the microplate reader should decline logarithmically, as you can see from the graph, which is inverted from your generation time assignment, which you've done quite recently. When groups are done, their plates, which eventually will be done in about for viable counting. You guys will place them at the top left incubator. Bear that in mind, folks. Make sure you've labeled your plates accordingly as well, all 5, and that you tape for all 6, and you tape them up accordingly. Thanks a lot. Don't forget, like, the labels, the practical, group letter, the date, the condition, the time point, exercise. in the bacteria as well. About once I've done this, so I would say about yes.
We're doing like an Instagram carousel of like targeted toward parents, wanting parents, developing. Basically, try your yeah, basically just like let your kids like. Yeah, like. Well, maybe not. Let your so that you pass it down to the ones. Oh. If you want to do first, you can talk.
Once the group is done, they can get ready for the presentation. To make it easier, I may not ask questions. If I do, they'll probably be simple. But at the very end, hopefully we should be done with due timing. I just want to highlight though, have a timer if possible on. to, for your FLR, give you sample data, because I want to make sure that you guys will be able to hopefully analyze what you would have expected for your results from the FLR. So check out Workus in about a week or less to see the results of what you could expect. And you could also describe limitations for your second FLR on what could have happened here. All who have done exercise for how many minutes are needed? You can share it if needed. How many groups are done exercise for? Raise your hand, you're done. Group C, group 3, are you guys done exercise 4? Groups 1 and 2? By the way, if you have already tried to start prepping up, we'll be getting shortly the presentations, and whichever group wants to begin first, they can. We'll be starting group 1 in 2 minutes. So just to confirm before we begin, any questions regarding exercise 1 to 5? So you should be able to see, observe, analyze, provide intro, thoughts, and discussion in 5 minutes. Try to even do it in 4 minutes, one per person, and don't worry too much, don't even worry about that. I just want you to be able to present the results that you have, how you would have expected, and what your implications are. So folks, we're gonna start now. I want you to be done in about 5 minutes. So let's try to get straight ahead. You can come to the podium. You can look at your results. You can talk about the results you have here. We're gonna start straight ahead. Exercise 1. Sorry, group 1. If you could come to the stage. Actually, groups, do you prefer to be at your tables or do you want to come to the stage? Okay, stay with the table. Like bench top. Bench top or podium? If you want to stay in your benches, that's okay. I don't mind. Yes, yes. And if any other groups wanted to hypothetically go first or change up the order, that's also okay. I just want us to be swift with timing. Can I also confirm everyone has given me their assignment, right? Practical that you were using like the L rods, the three tables, the glass pipette canisters, please return them back to the front. And we can begin. Is that okay? Folks, who wants to go first? Just to give time for those that maybe needed more time. Who wants to go first? 1, 2, 3, 4, 5, 6. Who wants to be first in the list? You're first? Group 2? Okay. Do you want to be at your bench? You want to be? Okay, okay. Okay.
Oh, sorry guys. Wait, do you want to stay at the bench? I just wanted to. I just feel like if we stay there, we're gonna have to like yell.
Everyone should hear you best wherever you may be.
We're already here, so okay. Hi
Folks,
guys.
are we paying attention? Okay, so I'm gonna start. Let's begin right now.
Okay. Just let me know.
You're right.
Okay, so today we'll be talking about our results for exercises 1 and 2, and we'll be talking about what we expected to happen versus what actually happened. So for exercise 1, we expected the UV light to damage the DNA on side A because it wasn't covered by the tinfoil. And for side B, we expected normal bacterial growth. For exercise 2, M. roseus, I don't know how to pronounce it, but whatever, is a UV-resistant bacteria that repairs DNA damage. So under light incubation, we would expect better bacterial growth due to photoreactivation, where enzymes use visible light to repair DNA damage. But under dark incubation, M. roseus would rely on nucleotide excision repair, which is less efficient than photoreactivation. I'll just be presenting the results. So for the exercise, looking at the lethal effects of UV, the plates that had that were separated into A and B, we actually saw like growth, similar growth on both sides. So this means that UV did not penetrate. We'll talk about like why that might have happened later. And then for the damage and repair exercise, for plate 1, which was under UV for 30 seconds and incubated with light, showed normal growth. So, just like a red smear. And then plate 2, which was also 30 seconds, but under dark incubation, there was no growth. However, we actually saw contamination. So like there was like fuzzy, translucent like colonies on them, which is not M. roseus. So that shows that possibly due to like improper aseptic techniques, that's why that happened. And then for plates 3 and 5, which were both light incubation and under UV radiation for 3 minutes, there was no growth. For plates 4, which was dark incubation, also under for 3 minutes, there was a normal red bacterial smear. And then plates 6 and 7 also showed the same.
Okay, so we just talked about how in the first experiment we didn't see any lethal effects of UV in either like the A or B side of our plates. So we believe this is because we did take off the lid, but then we put the plates upside down when we put them under UV, and so they still weren't able to be penetrated by the UV light because of the plastic layer. And so the foil originally was supposed to be the only thing covering the plates, but since we were afraid of like external contamination, which is why we put the plates upside down. But because of this, like normal growth was observed throughout the entire plate instead of just on one side. And then for the damage and repair plates, yeah, our second plate did get contaminated, but the rest of them, including our controls, they were all okay. They showed either normal growth, and like Marissa said, like our plates 3 and 5 showed no growth, which was a little weird because M. roseus is gram positive. And so we didn't expect it to be more affected than E. coli because none of our E. coli plates, which were under UV for 10 minutes, had inhibited growth. But our M. roseus plates, which were under UV for 3 minutes, did show inhibited growth. And so we thought for future directions, this would be important to see and probably like redo the experiment properly because in bigger labs, they do use UV as a disinfectant. And so it's important to know which bacteria it will work on and how long it takes.
And don't get shocked if Dr. Brown comes in and out. It's okay. You just present as you are.
Okay.
Don't feel like you have to continue.
So for exercise 1, we found that for B subtilis for both 7 days and 24-hour plates, they all had the most growth compared to the other bacteria, and we rated that a 3 for the range. And then for E. coli, since it's gram negative, as we expected, there wouldn't be much growth on there. And as for S. epidermis, there was like a little bit of growth. We rated that a 1. And we can conclude from this that the B. subtilis is more UV resistant because there was so much growth. But the thing was that there was growth all over the plate and not on one side. And we think that an error because of that was because we inverted them and we were supposed to keep them upright. So there was only supposed to be growth on one side, which was B, which was covered by the foil, and not side A, which was exposed to the UV. So yeah.
Okay, guys. So yeah, the reason why we think there's different growth, although we should have expected the parts of the sides B to have more growth compared to A, is because the gram-negative bacteria have a thin peptidoglycan layer, which is why they would be destructed by UV more compared to the gram-positive bacteria, which is S. epidermis and B. subtilis, 24 hours and 7 days. Okay. For the second experiment, we had plates, light plates 1, 3, 5, and 6, dark plates 2, 4, and 7, out of which we didn't get any growth. And out of our expected results would have been plate 6 and 7, which should have had growth because they weren't exposed to UV. Plates 1, 3, 5, and 6, with the Poisson lights would have had a little bit of growth on all of them. Plate 5, which had the lid on, should have not had, or should have growth as well, but we didn't have any of those results because we inverted the plates, even though we did cover them. But technically, we were supposed to have more growth on that plate because we had the lid on. Other than that, yes. Yeah, so for 1, it was because of the cell wall thickness, and for 2, it was also because of the light exposure with the glass lids on. So that's what it's. Thank you.
Sometimes if I have to briefly leave out with something changed.
have to sit up.
4 minutes, 1 minute.
Yeah,
per
we
patient.
will.
Okay, so for the first experiment we did, we wanted to we knew that UV radiation is detrimental to bacteria. So when the DNA absorbs the UV radiation with a wavelength of around 254 or higher, the dimers form, which distorts the DNA. We wanted to compare the gram-positive microorganisms and gram-negative organisms' response to UV radiation, as well as compare the role of endospores in survival with spore-forming bacteria in response to the UV radiation.
So for our results in the first exercise, we looked at both sides of the plate. Despite covering side B and exposing side A, we had similar growth on both sides, which wasn't what we expected. Even in terms of similar growth amongst the gram-positive and negative bacteria, and then the 24-hour and 7-day bacteria, because the gram-positive bacteria would have more of a peptidoglycan in their cell wall, which means that UV would penetrate less than the E. coli, which would have less peptidoglycan. And then with the B. subtilis, we would expect the 24-hour to have more endospores with a less effective UV. But again, it had similar to the 7 days, and then both similar on A and B sides. So again, in terms of, looking at these results, we can probably confidently say that it was due to the fact that they were inverted under the UV, not allowing the UV to penetrate correctly, but also possibly errors in plating because nothing worked out well.
For experiment 2, we looked at the repair mechanisms of Ambrosius. And so there's the light repair, which is photoreactivation and uses DNA photolyase which is activate under light. And it breaks the dimers and restores the DNA structure. And there's also dark repair, which is also called excision repair, where endonuclease cuts the damaged DNA and helicase removes the strand. DNA polymerase puts the new strand, and then DNA ligase fixes the strand.
Okay. And then for results, basically, plates 1 to 4 grew like nice colonies, and 5 to 7 did not. What we would have expected is for plates 1 to 5 to not grow because they were exposed to UV. We think that plates 1 to 4 grew because, well, the plates were upside down. There was a layer of plastic. But I did sort of notice that plates 1 and 3, the ones that were incubated in the light, grew like nicer and brighter. So, well, that would be because it's growing in the light, which it prefers. We don't really know why plates 5 to 7 didn't grow because 5 was exposed to UV, the other 2 were not. We don't think that that is what caused it to not grow. We think that probably we just did something wrong experimentally. And if we were to do this again, we would probably do it right side up so that the UV actually penetrates. And if you're afraid of like external contamination, you could do it in the biosafety cabinet.
So for the lethal effect of the UV lab, for the lethal effect of the UV lab, we divided the plate into 2 sections, exposed one side to UV for 20 minutes, and the other side covered by aluminum. We expected the side with the aluminum not to grow as much, and we expect gram-positive bacteria to grow more than the gram-negative because of their thicker peptidoglycan layer. But in all 4 plates, we see that both sides grew, most likely because the plate was upside down. So the UV didn't stop the growth on one of the sides. So in the future, we can aim to make sure it's the right way up.
Okay, so for the second part of the experiment, so the damage and repair, so
Yeah. So similar to the other group's background is that what we were doing was actually looking at the bacteria and seeing if it has mechanisms to repair the damage that we were exposing it to through the UV. And we were looking if they can actually do like light repair or dark repair. So in our experiment, we tested the Ambrosius under the different UV exposure conditions. And then we had certain expectations for all 7 plates, but we'll actually show you that our results for some of them did not show what we were expecting. But basically, plates that were exposed to shorter UV times, they should show some bacterial survival, whereas longer UV exposures would have significantly reduced that growth. And then plates that had that star-shaped mask, they should demonstrate that covered areas were protected from UV damage. And that would basically confirm like the direct lethal effect of UV. And then yeah, our control plates should just show like bacterial growth all over. So we'll actually show you the results.
So for the results of UV radiation and damage and repair. Okay. For plate number 1, we actually observed no growth, despite the fact that we should have growth in the unmasked areas and then like some growth in terms of inside it because when it undergoes photosynthetic repair under the light. But we observed none. For 2 under 30 seconds, we also observed no growth at all, even though we would expect some growth in the unmasked area again. But that should have less growth in the unmasked area for the first one, because that would have not undergo photosynthetic repair or activation. For plates 3 and 4, for 3, underwent 3 minutes of UV light radiation, and again we had no growth. Even though there should be growth in the unmasked areas again, and since it was under light, it should have photosynthetic reactivation, should have a little bit more in the unmasked area than 4. For 4, however, we had growth in the unmasked area and no growth in the masked area expected. For 5, we had full growth for the whole thing, and that's probably because of the fact that there's the lid on top, so UV light couldn't penetrate, so all the bacteria stay alive. And 6 and 7, no UV light that had growth. And then for 1, 2, 3, a limitation could for no explanation for no growth could be the fact that maybe we didn't even inoculate any bacteria to be killed by the UV light. Another thing could be like how there's no photosynthetic reactivation for 1 and 3. It could be the case where we have to factor in how long the incubation time under the light or that light intensity. And then we also have to take account the exact UV radiation intensity and the UV radiation distance of the light to look at why there is no growth in the place expected growth.
Lastly, for the antimicrobial susceptibility test, for the epidermis, we saw growth in all quadrants after re-inoculating from the zone of inhibition. So this means that it's basically all the antimicrobial, the antibiotics are all bacteriostatic since there's growth. And then in the for the epidermis, and then for E. coli, we saw growth in ampicillin, meaning it's bacteriostatic, but in the chloram, there is no growth, so it means it's bactericidal. But then for the streptomycin and the tetracycline, there's like dots, so it's a bit weird, but it could mean that the bacteria are like, some of them are resistant since there's like spreaded dots, or it's like persistent cells, so like they're in like dormant state. Like for the other two where there's like not a clear growth, it could be because these cells are like resistant or persister cells, like in like yeah. Yeah, which is weird that it's not like it could.
Can it be as a possible? Not really.
To be honest, I think that you have contamination going from this slant to here. Look, this is your inoculation. It went into this area. And same with here, it's connected. So there's issues with the way you plated your samples actually. Would you expect that ampicillin would be bacteriostatic?
Yeah. It doesn't cleave the beta, uses beta lactamase to cleave the beta strand. Yeah. Cell wall. Synthesis.
Bacteria that is on inhibition, we expect that to have been dead or alive.
Dead.
And just not grow. So what you're doing is you're taking the zone of inhibition where you saw no grow and ask, remember what you did. You inoculated the plate, you inoculated the entire plate. You put bacteria everywhere on the plate, right? And then antibiotic flat was. And then you drop the disc in each spot of your head. And the antibiotic diffuses out of that disc. And in that zone of inhibition, it's inhibited the growth of the bacteria. What you don't know from that assay is whether or not the bacteria in that zone of inhibition are dead or alive. All you know is they did not grow. Right. So the idea of the second part is to take the bacteria from the zone of inhibition and put it in fresh media and ask, do they grow? If they were killed by an antibiotic, are they going to grow? No. So you should have an idea of what should happen in this experiment. And if you're seeing that they're growing, then likely what happened, and think about how you did this experiment. So what did you have to do to inoculate this plate? You have the zone of inhibition. Find Okay. That doesn't work either. Time to get some new markers. All right. Okay, so let's say this is one of the divisions. You dropped your, and this is ampicillin disc, and you got a zone. Here you had nice growth, right? That's what you saw. I assume that's what you all saw. That's what you should have all seen. So the idea is we touch a tiny region here. And how did you do that? Describe to me what you did last week to that experiment. It's a cotton swab, yes. And the idea is you touch it, and then you put it on the plate. And the question is, does it grow or not? If it grows, then that tells you that the growth was inhibited, but not killed. But if you're not careful when you go to take the material from the zone of inhibition with that cotton swab, and you touch closer to here, you're going to touch some of the bacteria that you had that was growing. And likely that's why you got the result. Because the cotton swab is relatively big, and if you weren't paying attention to where you touched, even if you touched a tiny bit of the perimeter here, you were going to pick up cells that would grow, and they would grow quite readily. And that's likely what you did. Because we know ampicillin and penicillin are bactericidal. Right? They affect cell wall synthesis. The bacteria cannot grow on nutrient agar without their cell wall. So that means those bacteria had to be dead. Well, the other antibiotics affected what? Streptomycin, chloramphenicol, tetracycline. What did they affect? What step in the synthesis did they affect? They affect cell walls? What do they affect? Protein synthesis. And generally speaking, as we've also discussed in lecture, almost the majority of the protein synthesis inhibitors are bacteriostatic. They prevent the bacteria from growing, which allows then your immune system to clean out the leftover bacteria that aren't growing. But you also have to look at what your plate looks like. So if you're seeing things that don't make sense, then you start to realize, well, if I look at this and go, okay, if you look carefully, a lot of these have started to touch each other. You know, so this one is touching this one. So you streaked here, and then you came back and streaked here, and you would have pulled bacteria from the other area onto the plate. Okay, when you look, they're interconnected here, which means what you streaked here, and then you came back to streptocycline, you're going to bring the bacteria that touched here, and you're going to spread them as well. In other words, they weren't necessarily from what you touched, but rather what you put on the plate in your experiment. So you make sure your divisions are separate from each other. What is supposed to happen in photoreactivation studies? What is the purpose of putting things in white light? And why do we use two different arms to that experiment? Let's start with that question. Why use 30 seconds and 3 minutes? What would you might predict would happen if you go through the bacteria at 30 seconds versus 3 minutes? Under UV light. Yep. But keep in mind that you're not going to suddenly, so if that took place under the 30 second exposure, and you experiment the same way, you would still see bacteria in the, took the plate, one in white light and one in dark light. You're inoculated the same way. You would still see bacteria under the division of the dark light. What you can see is that the pigment may burst itself, just like the experiment is on the end of this PowerPoint. You can't get, if the bacteria is not growing at all, photoreactivation is not. If the bacteria has so many mutations that it's not growing dead, you can't photoreactivate. Photoreactivation will repair 1 or 2 genes and that's it. Okay, so if you saw no bacteria on the plate at 30 seconds that was put in the dark, how many people saw that? It actually left the plate uncovered when they exposed to the light. How many people had no growth at 30 seconds in the unmasked area? Well, I think I've seen that it exists. So, who had no growth in 30 seconds? Who had no growth? Okay, so then that was just this. There's a big amount of growth. Because nobody can do the star at all. So that's a lot. So you see the color. Okay, what about this? About 3 minutes or about 30 seconds? There are a lot of no. How many why do you have do they do on all 3 of them?
Well, that's 6 and 7, those are the control. Those are 6 and 7, those are the control. But
Why did they block up this?
that one was the lid on for foul.
Okay.
That one had to go.
And then these ones you had no growth.
Yeah.
So you can't interpret anything from this experiment because it's completely incorrect. Okay. And here we have growth everywhere. If we leave the lid on, we're going to have to explain that growth from bacteria. So do you think that's a legitimate reason? 3 minutes would kill everything on the plate. It was not protected. But where's that area that we got was masked? You just didn't inoculate it. You had a mask where it would have ended up with something like this. Right? So clearly this is not showing you the proper result. Okay, so there's something wrong with what you did in this experiment. It's absolutely no sense. In every class, 30 to 3 minutes in general speaking, 3 minutes kills everything. In 30 seconds, you do it correctly, maybe the cells to survive, it will grow as colonies. But the problem is if you take a lot of time to take your lids off and you start timing, you don't have 30 seconds anymore. Have something like a minute, 1.5 minutes. As a result, it kills everything. But though these experiments are completely wrong, they were clearly not done correctly. The only thing that you established in leaving lids on, which is why there was a control that had to be on, showed what? UV light cannot penetrate lids, can't penetrate plastic. So if you were to have seen what you would have expected, the 230 seconds would have a few colonies in the star area, the mass area. And you might have seen a couple of the colonies where white plates would turn to pink again. You look at the serration processes experiment, it's the end of this PowerPoint that you had access to before the lab started. Okay, so those of you who have chosen not to read and understand why you're getting the PowerPoints, to help you understand what you're supposed to do, these were supposed to be looked at before you ever step foot in the lab. Okay, you have to understand, you have to know how to interpret this data. You're going to have questions about data interpretation on your lab exam. You're given all the tools. Okay. Really something in terms of the way you set out class, you fail to do the experiments correctly. You should have growth everywhere. So you must have left a lot of lids on during the experiment. That was clearly delineated that that was not what was required for the lab. Yeah. Why would you do that? You don't say that you know. Why are they going through? What's the glass? It looks like it's supposed to go through all the egg. That was not right. That's not how you're supposed to do it. And if we're thinking logically, we say, why do we do this one with bacteria up? And why are we doing what we're doing with people that are? That was to prove to you that plexiglass protects against UV light. Why do you think sunglasses? Or made of plastic will block some light. Okay. If you're trying to radiate something in a biosafety hood, if there's plexiglass protecting what you're radiating, it will not work because the plexiglass blocks the radiation. So when you write your lab report up, you're going to have to explain why you got the results you did. They're totally explainable based on how you did your experiment. It will give an analysis of what you did wrong. That's what we do in science. When our experiment fails, we ask why, and we figure out what biological reasons to explain our results. And you have clear biological reasons. If you look at next lab, an experiment showed the kinds of results we expected. There's some issues with the way the cells that go out the plates, but they see what they're supposed to see. So what you established in large numbers is that UV light cannot penetrate plexiglass. Certainly not in the wavelengths we're using it at this point. Even if you light down, it's still limited to bacteria. Make sure you understand that you're killing and inhibiting. Same with today's lab. You affect the temperature, but there's also the killing effect. So, for example, you grow bacteria at 65 degrees. If the bacteria don't grow, does that mean they were killed by 65 degrees? How do we test whether they were killed by 65 degrees? Or what could we just do with the tube they didn't grow? You take that tube, this is, oh, it doesn't grow at 65 degrees. Let's see what happens if I put this tube at 37. And it grows. So was the bacteria killed? No, the bacteria growth was inhibited by the temperature. Now, in the case of E. coli, they will go 65 degrees cold, but that's how you would test it. So for the first half, it's the effect in your interpreting inhibition. For the thermal death time, you're actually asking which temperature killed the bacteria. In both temperatures, 60 to 65 kill. One temperature kill. If both kill, does one kill at a faster rate than the other? That's what the thing is. Your stuff. That's the lid upside down on a piece of paper. That's the lid. It says the lid. I know, I does not say that. It says put the lid on a piece of paper with alcohol on it. And why would you do that? Protect it from getting contamination. Don't put the lid this way. The bacteria can fall on the lid while you're doing the incubation. You have to read it carefully. All right. Continue.
So I really so folks, let's just take a moment. I know some of you may have to go, but let me just highlight a few important points. One is yes about the lid. I do want to highlight something important to note though. You guys weren't the only group, there were other groups as well impacted. So don't think wrong about yourselves that you were following the instructions. And to be honest with you, as Warren pointed out, it says place them and it doesn't specify. So don't feel like you guys did anything wrong. And what I will say is that to be very honest with you, UV should be able to penetrate. Because depending on the wavelength, I mean, we didn't know from the lamps themselves, right? But depending on the wavelength and the proximity of the lamp, and this was an issue from the past. I don't want to go through, I don't want to bore you guys, but the idea is that what I hope to do is, inshallah, give you guys some plates to post. I'll post them on Brookus, and you guys could use them for your FLR. I don't want you to think that like you haven't done your best, because from our previous experiences with each other, you guys done amazing, right? So I just want that to be recognized. And pretty much you've done your best. Just wanted to briefly highlight though, make sure that for all your plates that you've labeled them correctly, that you know where your mask is, where your in the case of the UV, but more briefly, being able to understand the different points that have been touched upon from our groups thus far. And I know for any additional questions and thoughts that you may be considering, could further be described. So what I mean is that for your intros, for example, some groups have pointed out about UV repair, the conditions, the E. coli or Staphylococcus epidermidis, and the repair mechanisms, and even generally speaking, the colonial growth of these bacterium, something that you want to consider. The common mistakes as well, aseptic technique, and in the case of essentially not placing your mask correctly and spread plating incorrectly can actually lead to inconclusive results. But that being said, you guys can go. Just make sure that you've cleaned up your benches. If you haven't given me your generation time assignments, please do so. Your unknown is down at that end. Once again, you guys keep it up. Keep up the amazing work, be inquisitive, ask questions when needed, but also try to essentially don't feel discouraged. Yeah. And actually, before we end off, so this is going to be part of your FLR, the formal lab report, your second one.
I believe it's due on the 2nd. Just confirm. I believe it's the 2nd of April. So I know some of you may be busy, but try to see what you can gather and obviously to replicate and hopefully start prepping up in advance with the material that you have from the results that I'll talk about, but you can also get ready your intro, the discussion with certain limitations, some extensions, you know, thinking about UV, thinking about your glasses, thinking about even being in a bench where some benches have UV added on top as lamps close by. Think about how that could hopefully impact bacterial growth. Before I answer. And if there's any questions, I'll try to answer them. I just want to also highlight, please keep up to date with the manual, the discussion questions, because this can also help you in the long run for your lab exam as well. Have an amazing week. Yeah. Yeah. Yeah.
But I know there was a clear confusion where all like the difference between Lidon and opportunity and also the place that you place down. Because of that, we have broken all the starship last time.
That's what I'm wondering. It's likely deleted some. So don't worry about the results.
Talk about how the starship map either due to air or just the fact that it's getting touch into the glass, like the plastic.
Can you repeat the last part?
You have those UV not going through the plastic, like the
sorry, folks, just to bear in mind, please follow through with the guidelines for the outline posted on Quirkus for the second and final, like your second formal lab report. Consider essentially the advice that I give in a week or so. I just want you to be able to understand the material and conduct yourselves accordingly. Make sure that you understand the components that we've talked about thus far and the points that Dr. Brent also highlighted, and you could see further in the manual and a bit more in the slides, including some responses to questions in the final slide from last week. So in terms, sorry, I don't want to detract, but your question was about essentially limitations and UV and essentially shape. Yeah, my goal is to essentially send you guys. For now, you can do it as you are, like describe the limitations. I think what would be nice is that you provide the limitations from your experiment, but focus your results primarily on what I give. So let's see. If you can, could you leave it at that? Yeah, it should be. I would recommend. Or you can send it to me a photo tomorrow, and then Thursday you can leave. That works. It is. Give me just one moment. 300. Okay. I can still look at it.
I really appreciate it.
Oh. Info procedure 1, 2, 3.
Can I see three again?
Do this.
You don't have to.
Okay.
I appreciate the work that you guys did with 5 and 6. Don't worry. Hopefully though some of the material connects and maybe you can make note of them.
Okay.
But apart from that, great job. Great job as well. Okay, I can look at yours as well.
This is mostly pushes. This week.
No problem. Take care. Have an amazing week, everyone. Wait, can I see two again? Okay. You ready? Okay. I'll give you your pen as well. One. That's okay. Okay. Thank you. No problem. Yes.
You write it as well. Because you're gonna do sound. Do we write it? But like, do we write it as we did it right? Or do we have to mention that we did it?
So I'm gonna talk about it. Essentially, do your results. Do your discussion considering the limitations of the of your plates, of your group. But I'll try to give results. Essentially.
Just like a small part, but the focus should be the interpret.
I mean,
just mention that.
Describe like the limitations, but provide it in detail. What I mean is like, okay, let's say there's this error. Talk about the error, what the significance of it is, and what you would have done to resolve it.
But the rest of the data is focused on the actual ones, right?
It's pretty, but it's not like as much as it appears. The results are limited because pretty much you have observations, the figures, and interpretations are left for discussion. So it's like in the meantime, you can begin the intro and maybe highlight some points describing them for your limitations. But I'll by that time hopefully give you.
Wait, was equal I?
Yeah.
So Brown was gaslighting me for nothing.
Don't worry. Don't worry.
What is?
Let me describe this. I'm sorry, folks. I didn't get to speak regarding your unknown assignment. So the idea is, okay, DT plus FC. So DT is your data sheet. FC is your flowchart. And essentially, if they were both complete accurately, then they would have gotten a full 1 out of 1. Now, the quality of the slides is something that I had to keep in mind. And that's essentially placed as percentages. And then I scaled the mark higher. So whatever you see on Quarkus is what you actually have. This is not, this is more like raw values, but they were corrected on Quarkus.
Because this 4%, there was like a formula. That's 1%. 1.5, 1.5? Because there was like a formula.
I think it was 3 or 4. 3. So 1.5 times 0.7 plus 1.5 times 0.7 plus that, and then I added a 0. Oh, so it looks, it's a bit different. But yeah, like do your best, folks. I just want to make sure that you're able to understand the material. And if I find that like things are, if there's things that I may have suggested, then hopefully you incorporate them accordingly for your upcoming assignments. But at the same time, try to see how you can best maximize your potential. Yes. 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 That's not maybe not be able to help considering the allowance. But Generally speaking, it could be a Bye, take care. You can. Okay. That's okay. That's inshallah. No. You still have it or not? Take care. Take care, both. Okay. Do you need the other one or no? Sure. Okay. So should I mark both or just out? It may be a bit difficult to find now. I'm just worried because